Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuh. All students, we are basically discussing the chapter 6 that was basically the enzyme. And if you remember, we have discussed these all topics. Like we have discussed what is metabolism, what is biocatalysis, what is catabolism, what is anabolism in our previous video, as well as we have already have discussed about the substrate, product, activation energy. So you should have to keep these all things in your mind and how the enzyme is basically lowering, or you can say how the enzyme is basically speeding up the reaction, or how the enzyme is lowering the activation energy. We have already discussed in the previous video. We have discussed about the intracellular enzymes and the extracellular enzymes. Now today we are going to discuss about the characteristics of enzyme. That is a very interesting chapter and you are going to love that. So before going in, let me to explain you that in 1878 there was a German physiologist named as Wilhelm Kuhne. Wilhelm Kuhne first used the term enzyme. What is now basically the enzyme? Enzyme is basically the globular protein. What is a globular protein? Basically, students, you know very well about the proteins, that protein is one of the most important component of the living organism. And there are further types of the protein, and one of the type of the protein is the globular protein. And the globular protein is what? Let me to tell you. The enzyme is basically made up of the linear chain of the amino acid, and that used to fold in a three-dimensional molecule. So you know it very well that the protein is made up of the amino acid. And you know it very well about the amino acids are basically the functional, or you can say that they are basically the units of the protein. The 20 amino acids are present in the living organisms, which are making the proteins of the body. So just like that, beta K carbohydrates are made up of glucose. Similarly, the amino acids or proteins are made up of amino acids. So there's a long chain of the amino acid, like the many of the amino acids used to join with one another, like through a peptide bond, which we are going to learn in detail in the higher classes. But right now, what you have to keep in your mind is that amino acids are basically thousands of the amino acids are basically a linear chain of the thousands of the amino acid. Now they are actually enfolding themselves. They are actually enfolding themselves into a globular structure. Globular. So this is basically a globular. So that's why it is said globular protein. They used to enfold themselves into a globular protein, globular structure. That's why it is said that the enzymes are globular protein. Three-dimensional molecule. 3D, you know very well. The 3D means that you can see it from three sides. So that is like that. Okay. Enzyme is basically a globular protein that is made up of the linear chain of the amino acid, and the amino acid chain used to enfold itself into a globular protein, just like that. But you have learned about the DNA, that the DNA used to enfold itself into a chromosome. Similarly, the amino acid chain used to enfold itself into the globular protein. Is that clear to you? So that's why it is said that almost all enzymes are protein. All enzymes are the protein, as well as they are made up of amino acids. So that is clear. Okay, the second major point is that basically beta, we know it very well that enzyme is basically speeding up the reaction. It used to speed up the reaction. Speeding up the reaction. So how we have learned in detail that it used to reduce the activation energy. So the enzymes, sometimes there are some enzymes, or most enzymes are basically making the reaction millions times faster as compared to the uncatalyzed, or uncatalyzed means the reaction we do not have any enzyme. So the enzyme presence make the reaction million times faster, as these catalysts or enzymes are not consumed by the reaction they catalyze. So to get the point, we are already from the start of the reaction, start of the chapter, we are learning that okay, enzymes do not utilize their self, right? They're only doing what? They're basically speeding up the reaction. So the enzyme is basically speeding up the reaction millions times faster as compared to the reaction which do not contain the enzymes. One thing about the enzyme which you have to keep in your mind that they are very, very specific for the reaction and for the nature of their substrate. That means that it is not like that any enzyme can speed up any type of reaction. It is not like that enzyme, any type of enzyme or any enzyme that is present, it can you can say it can catalyze or it can speed up any reaction. No, the enzymes are specific for the reaction. Let's suppose is a specific reaction. There is a specific enzyme might we have learned like protease. The protease is enzyme which is only acting on the protein. The cellulase enzyme is only working or only acting on the cellulose, right? So these are very like you can say that pepsin or like these kind of the you can say then I have explained you like pepsinogen. Pepsinogen is the enzyme which is acting on the protein. So these enzymes are very specific. It's not like that any enzyme can work on any type of the substrate. No, they are specific to the reaction as well as the nature of the substrate. It is not like that any substrate you can say that attach on any type of enzyme. No, it is not like that. This enzyme is very specific in the case of the substrate. Okay, one more important thing which I want to explain is that only a small portion of the enzyme molecule is directly involved in the catalyst, and that catalytic region is known as the active site, and it is recognized or here the substrate used to bind, and that is a place where the reaction used to occur. So it is like that only a small portion, only a portion of the enzyme is involved in the reaction. It is not like that this whole enzyme is going to involve. No, some specific portion is involved. Some specific portion is going to involve, and the specific portion is going to be known as the active site. And this active site is going to recognized by the substrate. Let's suppose this is the substrate and this is the site of the substrate which recognize the active site of the enzyme. So this whole enzyme is not going to take part in catalysis or in the you can say in the reaction. This part is going to take part which is going to recognize or where the substrate is going to bind, that is known as the active site. And this is basically the site where the reaction is going to occur and the substrate is going to convert into a product. Okay, the next point about the enzyme which you should know is about the inhibitors and the activators. Obviously, there are something in the body which is actually activating the enzyme and which is actually inhibiting the work of the enzyme. It's not like that once the enzyme is activated, then nothing in the body is going to stop. No, if this happens, so what will happen if the enzyme is going to remain activate? What will happen? We are going to the body is going to eat their own structure. The cells are going to eat, or the enzyme is going to eat their own structure. Their own part the body is going to dismiss. So it is like that there is something which is enhancing the speed of the enzyme, or which is diminishing like that, which is like finishing or stopping the production of the enzyme or like the working of the enzyme. Basically the enzymes are going to work according to the need of the cell body. Well, let's suppose a cell needs some specific amount of the enzyme. So this specific amount of the enzymes are going to activate. It is not like that uncontrollable enzymes are going to start. No, it is just like in a car. When you need to start the car, then you are going to start it. Or when you need to stop the car, you are able to stop. So similarly, there are the inhibitors which used to inhibit, which used to pause, which used to stop the work of the enzyme. And there are the activators which used to activate the work of the enzymes. So that's why it is said that Enzymes production can enhance or diminishing by the cell according to the need. Enzyme activity can also be regulated by inhibitor and activator. Fine. Okay. Some enzymes do not need any additional component to the work. This is very important, students. You should know about that. What is a cofactor? So for that, you have to listen me very carefully. Basically, students, sometimes it is happening that the enzymes are independent to work. Like they do not need any kind of the help from anything. Okay? But sometimes it happens that they need some known protein molecules or any kind of the ion for the work, and this kind of the ion will be known as a cofactor. Let's suppose, student, that there is an enzyme, okay, and for like this enzyme needs some additional protein molecule or any kind of the ion. Let's suppose it needs the any kind of the ion for the its proper functioning. For its proper functioning, it needs a specific ion. So this will actually help for enzyme to work better. Okay? And this ion will be known as the cofactor. Is it clear to you? So it used to happen that the protein sometimes needs some additional components like protein molecule or ion. So keep in your mind that the cofactor can be either inorganic. You know very well about the organic. You must have learned the definition of the organic and inorganic in chemistry. That organic molecules are those molecules which contain the carbon and hydrogen, while inorganic compounds are those molecules which do not contain any kind of carbon or hydrogen, like they do not contain the combination of the carbon and hydrogen. Like organic compound do contain the combination of the carbon and hydrogen. So cofactor can be of two types. It could be inorganic or it could be the organic. So the inorganic could be any kind of the metal like iron, copper, zinc, any kind of the metal. Or organic molecule could be like there are like Falvin, heme. These are bit of organic molecules which used to help enzyme to work actively. If organic cofactor are tightly bound to an enzyme, then that is known as a prosthetic group. That is known as what? That is known as a prosthetic group. If a cofactor, as I told you before, that there's an organic cofactor as well. If the organic cofactor is tightly attached with the enzyme, then that in that case it will called as a prosthetic group. And if the cofactor is loosely attached with the enzyme, then that will be known as a coenzyme. So coenzyme basically transport chemical group from one enzyme to another, and some important coenzymes are the riboflavin. These are the protein, thymine or the fulvic acid. So students, what we have learned? We have learned that the enzyme do not need any kind of the helping factor. Mostly the enzyme used to work independently. But sometimes it happens that some kind of the molecules or some proteins, or you can say that some ions used to help the enzyme to perform a proper function. Sometimes the enzyme needs some additional molecules. These are what? These are the additional molecules. Sometimes the protein or ions need additional molecule for their proper working. And in that case, these additional molecules are known as what? They are known as cofactor. Now, cofactor can be of two types: organic, inorganic, or organic. The inorganic are basically those cofactors which do not contain the combination of the carbon or hydrogen, or in that case mostly are the metal ions. So the metal ions could be zinc, could be iron, could be copper. While the organic have further two types, like if organic if it's tightly attached, if organic is tightly attached with enzyme, very tightly. If organic cofactor is tightly attached with the enzyme, then that will be known as a coenzyme. If it is tightly if it is tightly attached with the enzyme, then that will be known as a prosthetic, not the coenzyme. I'm really sorry about that, student. That is, in that case, that will be known as a prosthetic. So that if the organic compound is going to be tightly bound with, if this organic cofactor is tightly bound with the enzyme, then that will be known as a prosthetic group. And if it is going to loosely attach with the enzyme, then that will be known as a coenzyme. The most common example of the coenzymes are the vitamins. Like some common vitamins are the you can say flavin, thiamine, folic acid. These are some coenzymes. These are some vitamins which are acting as a coenzymes, that when the enzyme needs that organic compound, these organic compounds are going to attach with the enzyme, and enzyme used to get activate. While the prosthetic group, they are tightly attached. These cofactors are tightly attached with the enzyme, and the examples are beta heme. Okay, hemoglobin, you might have know that heme is basically helping to form the hemoglobin that is present in the blood. So hemoglobin is basically the protein. Okay, so that is a heme. Or there are also like other examples are there, which is going to discuss in your book, as well as we are going to discuss in future as well. So that is all about the enzyme. That we have learned that enzyme basically used to work independent, but sometimes it used to happen it needs some kind of the additional component, and these additional components are known as the cofactor. And the cofactor, if the inorganic additional component is going to attach with the enzyme, then that is basically known as like it could be the methylene, it could be fire, you can say that ferrum or the copper. But if in the case of the organic component of the enzyme is also there, organic component is also their cofactor, and if it is going to tightly attach with the enzyme, then that will be known as a prosthetic group, and if it is going to loosely attach, then that will be known as a coenzyme. So hope so you get that. Okay, student, this is most interesting point, that several enzymes can work together in the specific order, corrugating metabolic pathway. It's like that, student, that many enzymes are working together. They all are working together to make a one product, and especially the example is the metabolic pathway. In the metabolic pathway, one enzyme take the product of another enzyme as a substrate, and after the reaction, the product is passed on the next enzyme. Did you get that? Let me tell you. They are saying that beta, there are many enzymes. Let's suppose there is an enzyme like A, B, C, D, like let's suppose these all are enzymes. Okay, now these enzymes are working in the series. These enzymes all are working together to produce the final product, and that is known as the metabolic pathway, where many of the enzymes are going to involve. And the metabolic pathway is basically best example which is expressing that several enzymes can work together. Let's suppose, student, there is a substrate A that is converting into the B product. It is producing B product, and this B product is going to be a substrate and will produce the C product. And this C product is going to produce this C product is going to be a substrate and will produce the D product. Now listen to me. This is a substrate of the enzyme A and will produce the B product. This small b is basically the substrate of the enzyme B and will produce the C product. And this C product is going to be the substrate of the enzyme C and will produce the D product. Right? And this D is going to be the substrate of the enzyme D to produce the E product. So what is happening, student? That these enzymes are working in the series, right? They all are working together to produce the final product, that is a E. But what happens here? Listen to me very carefully. One enzyme take the product of another enzyme as a substrate. This is the enzyme. This is taking the product of the enzyme A as a substrate. In this reaction, the B is the substrate, but in this reaction, the B is the product. In this reaction, the C is the product, while in this reaction, the C is the substrate. In this reaction, the D is the product, while in this reaction, the D is the substrate. So in that case, one enzyme takes the product of another enzyme as a substrate, and after the reaction, the product is passed to the next enzyme. So I think so that you get my point very carefully. So student, if you're not getting the lecture, you have to see the video again and again and try to make the beautiful notes, the perfect notes. It will enhance your vocabulary. It will also help you to write by your own. Okay, so work on this strategy. Inshallah, in next class we are going to learn about the usage of enzymes. But student, keep in your mind that this topic 6.1, the characteristics of enzyme, is a very important topic because it is discussing the characteristics of the enzyme, and these are the basic characteristics which is going to move with you till your master's level. Okay, so work on it, and if anything is not clear to you, you can just comment me and I will explain you. Okay, best of luck and Allah Hafiz.